Hello, everybody. It's a little weird for me to think, is this good afternoon or good morning or is it good lunch? Or so when the joint seminars were at 4, it was easy, good afternoon, right? So welcome, everybody. What we're in for today is a special treat. It's the Eiduson lecture, which will be given by Tessa Harrison. The Eiduson lecture is given in honor of, usually we have a little picture here of Sam Eiduson, so it's given in honor of Sam Eiduson, and for those of you who don't know who Sam Eiduson is or was, he actually was a very influential person at UCLA. He started the Neuroscience IDP back in 1972. He was the first director until 1985. So he started, for you graduate students, this is the reason why you're here. It's actually for Sam Eiduson. He inaugurated this lecture series if you believe in a biography of his, in 1982. If you go with the BRI website, this started in 1993. So in either case, it's quite a long time ago since we have these lectures to honor particular student who has done very well in this session. Just a couple of words to Sam Eiduson. He was born in 1918, then did his undergraduate work in chemistry here at UCLA, participated like many people at that time in the Second World War. He was mainly in the Pacific, and then came back to UCLA and got a PhD in chemistry. Stayed on at UCLA and got a faculty appointment in biological chemistry and psychiatry. So he was one of the first people that actually really valued and lived interdisciplinary research. His main work was very interdisciplinary at the time, as he was investigating molecules that would both change the biochemistry of the brain as well as change the mood, the behavior of the brain. So he was very interested in biochemistry and behavior go hand in hand, and he was one of the 4 authors of the first textbook, biological chemistry that was published with him as a first author. He worked first on decarboxylases and then on monoamine oxidases. So he's been very biochemically influenced. So I thought I was going to introduce Tessa, but then it turned out that Susan Bookheimer, who is her mentor, is actually here. And since Susan knows Tessa a lot better and knows her research a lot better, I'm going to give my microphone over to Susan.
Thank you, Felix. Thank you all for coming here. It's a great pleasure and an honor for me to introduce my terrific graduate student, Tessa Harrison. And she's extremely well deserving of this fantastic honor. Tessa graduated from Georgetown University, where she was a major in neurobiology, a minor in psychology, 2009, and she did research then studying dopamine receptors in several mouse models. She then went to spend 2 years as a research assistant with the great Marcel Mesulin in Chicago, and her work there resulted in just over 2 years in 5 papers, including one as a first author. We were happy to have her come to the NSIDP in 2011, and she joined my lab in 2012. And she's continued her work here in the aging world, focusing on genetic risk for Alzheimer's disease and its relation to structural and functional brain imaging. And she'll speak to you about her work today. She will be defending her dissertation this summer, so she's gone through very quickly. She's been incredibly productive since she's been here. She's been an outstanding graduate student and well deserving of this. Her record really speaks for itself. She's an ARC scholar. She won an NRSA. She's won numerous travel awards. She's already has 2 first author papers published since she's been here, and she's got another one under review and several others in the what would you call churning right now, but should be out before July when she leaves. But in addition to that, Tessa has also been a really great citizen at UCLA. She was very active in first developing and then teaching the neuroscience methods class, which is now required for NSIDP students. She's co-coordinated the neuroimaging affinity group with the BRI and also coordinated the neuroimaging journal club. She has been an avid presenter of her work. She's actually made over 17 poster and 6 oral presentations over her career. And she really does present herself well. She's a model mentee. Here she is at her outstanding dissertation proposal meeting. And she impressed her committee tremendously with just how well organized she was and how carefully she's planned her work. She's made great presentations, including this one at the Organization for Human Brain Mapping in Hamburg, Germany, in 2014. And then the year after, in 2015, in Honolulu, she made another outstanding presentation. And I have to say, as a mentor, it's really gratifying to feel like you're having an impact on your student. So, with that, I would like to very happily introduce my student, Tessa Harrison.
Thank you, Susan. I had a feeling those pictures were maybe going to be involved, but I was forbidden from opening that little slide set, so I didn't know for sure. Thank you so much for the introduction. That was really nice. And thank you all for coming. It's really nice to see so many familiar faces, and especially so many students. It makes me feel a little better. I've attended this talk every year since I came to UCLA, so it really is a great honor to be giving it this year. So the title of my talk is Neuroimaging Markers of Genetic Risk for Alzheimer's Disease in Cognitively Healthy Cohorts. And I want to start today just by talking a little bit about why the research I'm going to describe is important. So everyone here, I'm sure, has heard a little bit about the global aging population. And in fact, predictions indicate that quite soon the number of people aged 65 and older will be greater than the number of people aged 5 and younger for the first time in human history. And this is just one of dozens of potential images I could have used to kind of illustrate this point. And another way to think about it that is a little more helpful, I think, is to use these distribution plots that are often used in demographic research. So if you move from left to right here, this actually, the change in the shape of these plots is described as the demographic transition. And for our purposes here in doing biomedical research, it's probably more helpful to think about this based on a companion theory in epidemiology called epidemiological transition, in which each of these stages has a particular definition. So the first stage is called the age of pestilence and famine, which I love because it sounds so dramatic and dire. And this is a stage marked by resources that ebb and flow, high rates of infectious disease, and high infant mortality. We move into the age of receding pandemics, where advances in science and technology and the stabilization of basic resources leads to this decrease in infectious disease mortality, and especially a decrease in infant mortality, and you see this skew towards younger age groups. And then finally, you get to the age of degenerative and man-made diseases, where you further advances in science and medicine, public health awareness, other things cause this evening out of the different age bins and age distributions, and the main causes of death and mortality are the degenerative and man-made diseases like type 2 diabetes. And you can see this if we look on the left here in middle-income countries and on the right in high-income countries. On the left, Alzheimer's disease doesn't even make it to the top 10, the list of disease burden. But in high-income countries, Alzheimer's disease and other dementias is ranked 4th on the list of the top 10 diseases that cause the highest burden in the population. And so in the United States in 2015, there were 15% of our population was 65 and older, and about 5 million people had dementia. In 2050, these numbers are going to go up, with 21% of our population being 65 and older, and 15 million people suffering from dementia. And if you look at the global numbers, these are even more dramatic, with well over 100 million people suffering from dementia in 2050, in the absence of a preventative strategy or a therapeutic strategy between now and then. But of course, dementia is just an umbrella term, and Alzheimer's disease is the predominant cause of dementia. Dementia can also be caused by vascular disease, dementia with Lewy bodies, primary progressive aphasia, frontotemporal dementia. But if we zoom in on Alzheimer's disease, we know from twin studies that it's highly heritable, with 60 to 80% caused by genetic background, and the rest due to environmental factors. Alzheimer's disease genetics has a relatively long history, study of Alzheimer's disease genetics, with a landmark really being in 1993 when ApoE was first associated with the disease. The ApoE risk allele, ApoE epsilon 4, is still pretty unique in the world of polygenic neurological disorders. We also know that family history, sex, Alzheimer's disease is a sexually dimorphic disorder with twice as many women being diagnosed as men, and other risk factors, other genetic risk factors, all contribute to this genetic component. There also have been several rare variants found that have been associated with Alzheimer's disease incidence. These generally have slightly higher effect sizes. And then, of course, the number one risk factor for Alzheimer's disease is advancing age, which has both an environmental and a genetic component. Now, if you look, the Alzheimer's disease genetics is a little bit even more complicated because there are these dominantly inherited familial forms of the disease. With these forms of Alzheimer's disease, there's either a mutation in APP, which is the amyloid precursor protein, which is the protein that is then processed into the amyloid, which then forms the amyloid plaques in the disease, or presenilin 1 or 2, which both encode enzymes that are part of that processing, of the amyloid processing pathway. In a Swedish cohort, it was also discovered that a duplication of APP caused dominantly inherited Alzheimer's disease, and overexpression of APP is also believed to be the reason why people with Down syndrome almost invariably get Alzheimer's disease at a very young age, because APP is located on the 21st chromosome. But what's really nice about this picture is that this all points to amyloid. It all supports the amyloid hypothesis. It points to one molecular pathway. But when you look at the much more common late onset sporadic Alzheimer's disease, the list of associated genes does not look nearly so pretty. These are genes of varying ontologies that are not all pointing to one pathway and really serve to illustrate how complicated Alzheimer's disease is and how many different molecular mechanisms are contributing to the disease, from amyloid to tau to neuroinflammation and others. Several of these have been discovered in recent years with the advent of genome-wide association studies. The largest genome-wide association study published on Alzheimer's disease so far came out in Nature Genetics in 2013, and it was actually a really encouraging moment because they were able to replicate all of the previously identified risk variants in the smaller but well-powered GWASs, and also identify 11 new loci. But so why is Alzheimer's genetics and understanding the genetics of underpinnings of Alzheimer's important to actual disease prevention? So to get at that, I want to first look at this hypothetical model of Alzheimer's pathogenesis. So this is a model developed first by Clifford Jack and now sort of refined by lots of different groups that shows different Alzheimer's pathological processes and symptoms and plots them over time in a hypothetical order. So plotted in red here is amyloid or A-beta deposition over time. And you can see that the change, or the beginning of the change in amyloid deposition moving from normal into an abnormal range happens long before any clinical changes happen, which is plotted in that dark green. So it's this gap between the earliest pathological changes and the emergence of clinical symptoms where you might go see your doctor that is the increasing focus of this field. We need to better understand what is happening in this preclinical phase and how do we identify people in this phase. And if you apply like a magic cutoff here to all of these different markers where below is normal and above is abnormal, you can begin to see that you could potentially be defining preclinical stages. And then this is where genetics really comes into play. These people in these preclinical stages are healthy, happy, walking around, living their daily lives. And so genetic risk, which is quantifiable and doesn't change over the lifespan, is one way that we can identify people who should be involved in more thorough, expensive investigations like PET scans, spinal taps, and other in clinical trials that are getting at this phase, this preclinical phase of the disease. And this leads directly into what I sort of believe the role for neuroimaging genetics in Alzheimer's prevention is. Again, this characterization of this preclinical phase of the Alzheimer's disease, which must happen in parallel with all of the labs and companies that are working to develop novel drugs or repurposing drugs, or even working to develop behavioral treatments for Alzheimer's disease. But it also has an important role to play in clinical trial design. The pre-screening methods that we should be using and have started sort of to use in clinical trials are can and should be based in neuroimaging genetics research. And then, of course, matching your cohort and making it as homogeneous as possible with your treatment strategy is, I think, the only way we're going to ever achieve effective prevention in this disease. So finally, these are the big questions in the field. Can neuroimaging methods be used to identify structural and functional brain changes that predict AD in the preclinical stage? Will genetic risk factors provide predictive power to identify healthy individuals who are at greatest risk for AD? And then finally, is there a benefit to a neuroimaging genetic combined approach that takes advantage of both genetic risk and endophenotypes, neuroimaging endophenotypes for Alzheimer's? So these questions form sort of the scientific basis of the studies I'm going to describe to you guys today. I just went through a little background, and I want to go through the outline of the rest of my talk. So the first two studies I'm going to describe, the participants in those studies were older adults who were cognitively healthy. In the first one, we looked at genetic risk scores for Alzheimer's disease and hippocampal morphology and thinning over time. The second one, we looked at memory-related connectivity in ApoE4 carriers, and again, that's that risk variant of the ApoE gene. The last study I'm going to describe is a very exciting study. We're really excited about how the results have turned out so far. And the participants in that study are college-aged, and in that we looked at resting state network architecture, again, in ApoE4 carriers compared to non-carriers. And then I'll finish up with a little bit of implications of this work and some future directions. So diving right in, we predicted that a genetic risk score for Alzheimer's disease would be associated with thickness of subregions of the hippocampal complex, so the whole medial temporal lobe. And we further predicted that thinning over time in those subregions would be also correlated with genetic risk for Alzheimer's disease. So before I jump into the specifics, I want to just again remind everybody, so Alzheimer's disease is unique amongst these polygenic neurological disorders because there is a risk factor that has a moderate effect size, and that's the ApoE4 allele. And that's why studies designed to identify risk groups and non-risk groups based on just ApoE are feasible. But most risk variants that have been associated with Alzheimer's disease and other polygenic disorders are extremely common and have a very low effect size on the incidence of the disease. So here in this image, you can see CLU is an example of one of those genes. It is a very high, the risk variant, it occurs in over 80% of people, and the effect size is very small. So it makes it very difficult to study CLU in isolation. So there's increasing rationale. If we want to actually capture everything we've learned about Alzheimer's disease genetics, we have to take a multiple loci approach or a polygenic approach. But I've seen a lot of work, especially at conferences with null results, that take everything that's above that red line, put it into a score, and then test it with a variety of different AD biomarkers. And I think that, again, as I said earlier when we were looking at those lists of genes, this isn't really the best way to go about it because there are a lot of different processes happening here, and I think you're actually putting a lot of noise into your risk score if you include all of them. So it doesn't really make sense that each of these risk variants would be affecting each biomarker for Alzheimer's disease. So some may affect hippocampal integrity because they might be involved in signaling there. Some may affect amyloid deposition, etc. So for our study, we wanted to focus in and be a little more hypothesis-driven. So we used a two-level selection criteria to create our score. One, that they needed to be associated with Alzheimer's incidence, of course, and then two, there needed to be evidence in the literature that the risk factor was associated with either hippocampal structure or function, because that was the biomarker we were going to look at. So when we surveyed the literature, we saw ApoE, CLU, and PCOM all fit that criteria. And we also decided to include a positive or negative family history for the disease in our score because there's a rich literature showing that it is associated with hippocampal integrity in older adults, and also it kind of serves as a proxy for other genetic risk factors that we don't have that evidence in the literature to support. So once you know what you want to include in your risk score, how do you create a metric? So the easiest thing to do is to take an unweighted sum of the risk alleles or factors. You can weight those alleles or factors by odds ratios or some other statistical metric. You can look at genotype patterns and epistatic effects, or you can do gene-based tests where you look at groups of SNPs on a single gene. The latter two methods, you need really, really large sample sizes to make those be effective because you're binning people into such specific categories that you get very small groups very quickly. So we took the first two approaches, And we wanted to do both of them because we really were interested, is there actually a benefit to one approach over the other? So we created the scores. The unweighted score varied from 0 to 7 and weighted from -0.18 to 1.25. And again, those were weighted by the logarithm of published odds ratios from that GWAS study I showed you, and for the family history from a meta-analysis of the literature. Again, related to Alzheimer's risk and also the hippocampus in humans. So the hippocampus, of course, and adjacent cortex are some of the earliest affected regions in Alzheimer's disease. And in our lab, we've been using this high-resolution structural scan to maximize our ability to segment out the different subfields of the hippocampus, where we take our slices perpendicular to the longitudinal axis of the hippocampus and then use that data to create these fine segmentations of the different subfields. Because of the early susceptibility to Alzheimer's pathology, we thought this would be a good place to look for an association of increased genetic risk and thinning over time. Really briefly, our image processing steps involve manual segmentation of the white matter in hot pink in the upper left corner, and then delineation of the edge of the gray matter ribbon in the teal color. We then use anatomical boundaries, or anatomical landmarks, to create boundaries between the different subfields, and you see in the top right in B. And then we use an algorithm to take that gray matter strip that you see in A and actually unfold it into two-dimensional space. And then those demarcations are extended to form boundaries, and those boundaries are then used to create ROIs. Those ROIs are then mapped back into in-plane space, and thickness measurements are estimated from there. Our cohort for this study, we had 66 participants at baseline, and we had follow-up data after 2 years for 45 of them. Here I'm comparing our baseline and follow-up cohorts because I wanted to make sure there weren't any biases across the groups that would affect the association that we see with genetic risk score in either of the two cohorts. All of our subjects were normal, had normal cognition for age, and a minimum MMSE of 27, which is a pretty high standard actually. If we just look first at the genetic risk score distributions, again, on baseline participants on the right and the subset that had follow-up on the left, we see that the distributions don't differ across the two groups. And for unweighted risk score, which is on the top, it's actually pretty normal. On the bottom, there's a little bit of a skew. So I did some non-parametric tests, and our results were actually unchanged. The first thing we did is we took that baseline group, and we looked at the whole hippocampal complex thickness. But of course, there are natural variations in size of the hippocampus, so we had to control for that somehow. So we use intracranial volume to normalize each subject's whole hippocampal thickness estimate. And then we looked for an association with our two different genetic risk scores. And we see that there's no significant association between risk score and baseline hippocampal thickness. We also ran multiple regression models controlling for age and sex to make sure that we weren't just, that there wasn't a confounding factor that was not allowing us to see this association, and we did not see anything. We then looked at renal cortex specifically and subiculum specifically because we hypothesized early on that those two regions would be amongst the most vulnerable, and still saw no association. So before I get to what we saw when we looked at change in thickness over time, I just wanted to show you this spaghetti plot. This is each of our 45 subjects and their change in thickness, raw thickness over time. And this is just kind of a proof of concept that our approach was working. This is in line with what has been shown in the literature in terms of the magnitude of change over time in about 2 years. And when we look at genetic risk score, again, unweighted on the left and weighted on the right, and whole hippocampus percent change in thickness, you can see that as risk score increases, the thinning that a person experiences over time also increases. When we look specifically in entorhinal cortex, we saw that this effect was particularly strong. And in that subiculum, and again, these two we were most interested in because they're two of the earliest subfields affected by Alzheimer's pathology. We could see in the subiculum that the effect was still there for the unweighted risk score, but it was just a trend for the weighted risk score. And then again, I mentioned earlier that we can do this unfolding process where we look at a two-dimensional view of the hippocampus. So this is that unfolded map. It's at the anterior part of the hippocampal complex towards the top and the posterior towards the bottom, and then moving lateral to medial, left to right on the bottom. And you can see that the only two regions that didn't have a statistically significant association with genetic risk score were CA23G and the perirenal cortex. So for this study, we were excited that we were the first group to be able to show that a genetic risk score for Alzheimer's disease is associated with thinning over time. We compared our model with our risk score both risk scores actually, to a model that had just APOE alone or just family history alone, and the risk score really does perform better than either of those factors alone. We were excited to see that the relationship was strongest in entorhinal cortex, because this is in line with kind of the theory of Alzheimer's progression across the brain. We did not see any association in the cross-sectional data, which I think highlights the need for longitudinal imaging data when you're doing studies like these. And even I didn't show this data, but there was no relationship between genetic risk and cognitive scores and memory performance in this cohort. And that's when we enrolled cognitively healthy individuals. And this, again, serves to illustrate that sort of soft endpoints or paper and pencil cognitive tests are not really going to be the best endpoints for when you're studying groups of people who are cognitively healthy. And finally, our unweighted additive risk score was at least as effective as capturing risk as our weighted risk score. And this is important because for each genome-wide association study that comes out, odds ratios change slightly. And so if you want to implement a protocol across clinical trial sites or across even just research sites, being able to use something that does not change over time is advantageous. Okay, so moving on to the second study we talked about today. The participants, again, in this study were similar to the last one. They're older, cognitively healthy people who, in theory, might be in that preclinical phase of Alzheimer's disease, but we don't know. And we looked here at memory-related connectivity in APOE4 carriers. We predicted that our cognitively healthy older adults who carried the APOE4 allele would show deficits in cortical recruitment during a memory task compared to non-carriers. And again, here we're looking just at APOE and defining our groups by by APOE genotype. And that's because, unlike the previous study, we didn't have a strong hypothesis about other risk variants and functional connectivity. And in fact, there still is a lot to be learned about APOE's effect on functional connectivity. That was the focus here of this study. And before I talk about the study in specific, I want to just give a quick background on functional MRI. We were talking about structural MRI before. This is functional. So in functional MRI, we capture an image of the whole brain, which of course has 3 spatial directions. But then we repeat this every 2 seconds for the duration of the scan, which gives us a 4th dimension, which is time. We can then look at a particular spot or point in the brain and look at how the bold signal or the blood oxygen level dependent signal varies in that point in each of our images or over time. We can then plot this out as the time series. And one thing we do with people is when we put them in the scanner and give them a functional MRI scan, we give them a task to do. And so then we can look at how that time series varies with the task. And so you can see here in pink, the time series, that's the bold percent signal change. It's tracking fairly well, it's a noisy signal, with the task in this case, which is a simple eyes open, eyes closed task. And so you can do things like subtract out the closed phases from the open phases and find regions where blood flow or the bold signal were significantly different in the 2 phases. And in this simple eyes open, eyes closed task, you would see that there is significant difference in blood flow in the visual cortex. So then, taking this one step further, what's functional connectivity? What do we mean when we talk about functional connectivity? It's essentially just the coherence or the Pearson correlation coefficient between 2 of these time courses in 2 different regions of the brain. So you can either look at a voxel or at a region of interest. You plot the time series over time, and then you want to you use Pearson correlation coefficient or other metrics to essentially rank the similarity of the different regions you're interested in. And in the context of task-based fMRI, there's a particular method known as Psychophysiological Interaction, which gets at this idea of functional connectivity while integrating the fact that you have someone in the scanner performing a particular task. So I'm going to step through this in a hypothetical way. So assuming we have a memory task and we're interested specifically in the encoding phase of that task, which is plotted in the dotted line. We then take into account the delay in the bold signal after the actual presentation of the stimulus, which is the smooth line. Then, for a PPI experiment, you want to choose a region of interest or a seed that tracks really well with your phase of interest. So, for an encoding experiment, you might believe that the hippocampus, blood flow to the hippocampus is going to track well with the encoding phase versus the non-encoding phases. And then what PPI allows you to do is identify regions of the cortex, or other regions in the brain, depending on where, if you limit it or not, that are specifically coherent with your seed during that phase of the task you're interested in, and not during the phases of the task that are non-active or baseline. So here you can see, if I'm calling the red line retrosplenial cortex, it's out of phase and anticorrelated with the hippocampus on the far left, and comes into phase during the encoding block. And so we use a general linear model to essentially tease this out and look for regions that are significantly different, where the functional connectivity is significantly different in the context that we're interested in. So specifically in this study, we looked at a unrelated words paired associates task for people. It was a pretty easy task because we're giving it to older people. We want them to be able to complete the task. They just learn pairs of unrelated words like jazz and beast. There's an active baseline where they have to press a button every time the symbol changes, and that's to prevent them from rehearsing the words. And then there's a retrieval phase where they see one of the words, and they're asked to recall the second one or its pair. And they repeat these blocks several times. And then in this study, we looked at actually two different seeds, the anterior third of the hippocampus and the posterior third of the hippocampus. And we did this because, well, we didn't look at half and half, because when we take it from this really high resolution structural space up into functional space, which is lower resolution, we didn't want to have any bleeding between our ROIs. And we wanted to look at the anterior-posterior functional segregation, because there is a literature that says that anterior regions are more involved in encoding processes and posterior regions are more involved in retrieval processes, and we want to be able to see that if we could. Our cohort started out with 93 subjects, aged 55 and older. And in the process of reviewing, or being reviewed, we actually excluded 9 subjects because our reviewers wanted a more specific genotype group. So we ended up with 34 carriers who were all E3, E4 genotype, and 46 non-carriers who were all E3, E3 homozygous. So we had one difference in cognition here where the carriers were actually performing a little bit worse on verbal memory, and so we went ahead and included that as a regressor of non-interest in our PPI analyses. So again, just to review, we have 2 task phases and two regions of interest, which creates 4 potential conditions. So if we zoom in on one of these conditions, the anterior hippocampus during encoding, we can ask many things, but 2 very simple questions. Are there regions that are more connected to carriers and non-carriers, or non-carriers over carriers? If we focus in on one of them, we're asking specifically, during encoding, are there any cortical regions that are more functionally connected to anterior hippocampus in non-carriers compared to carriers? And again, our prediction, as I said earlier, was that regions that the non-carriers were recruiting to help with this memory task would be blunted in our carriers. So we can see this lower interaction effect. And indeed, we saw regions that were different, including here the precuneus regions in the precuneus insula and cingulate, but our results were not exactly what we expected, because if I put that orange line right at 0, what we're actually seeing is that in the non-carriers, there's this positive interaction effect, and in the carriers, it's a negative interaction effect. So it wasn't even that it was a blunted effect, it was that there was this actual uncoupling of the hippocampus from these cortical regions that the non-carriers were positively coupling to. And if you look across, that we see that same direction of magnitude of difference in our other regions, and indeed it's consistent across the different conditions we looked at. And now we could dive into this, but I think the most interesting part about this study was this negative PPI. So this is very busy, but when we saw this pattern across our different conditions, we looked just specifically now at regions where there is a significant, and this is FDR corrected at 0.05, a significant negative interaction, so again, uncoupling in our 4 different conditions. And you can see that for the carriers, there's lots of regions, especially in anterior parts of the brain, where you see this uncoupling effect. But for non-carriers, which are shown in red, there's not very much of this effect. They're pretty much only in the posterior hippocampus encoding phase. So this widespread, significant hippocampal cortical uncoupling during a memory task, we hypothesized that this might be based on more of a network difference, a network level difference, and we're not able to, and we need to kind of move up a level to see it. So now I'm going to take a minute to just introduce the idea of intrinsic connectivity networks. So probably most of you have heard about the default mode network, which is shown here in red, which is a network that is relatively more active during rest, so when not engaged in a particular task. And the medial temporal lobe is actually an interesting part of the brain in that it is sometimes coherent with the default mode network, and that is thought to maybe underlie memory consolidation. So when you're not actively involved in the other tasks, part of the memory consolidation process is happening. And then, of course, when you're actively engaged in a memory task or trying to remember something, the hippocampus is going to be uncoupled from the default mode network and be functionally coherent with memory regions. And it's actually been shown in Alzheimer's disease that this switching is actually a problem, and that the switching doesn't happen as cleanly as it needs to, and that may underlie some of the early memory problems. So in this study, this consistent magnitude and direction of the interaction effect that we saw to us suggested that there was a network difference going on that we would need to explore further in resting state functional data in these subjects. And as I already said, the inability to modulate default mode activity when you're supposed to be actively engaged in a task is actually a feature of early Alzheimer's disease. So we think that problem switching the hippocampal coactivation with default mode network and then with memory retrieval networks or encoding networks during a task may underlie these findings. And if you want to read more about this, you can read the paper that we published in HBM last year. So, last one. In this study, our participants, again, were college-age adults, and we were interested in looking at those intrinsic connectivity networks that I just briefly introduced and how the architecture might be different between carriers and non-carriers with the APOE4 allele. Our prediction was that the carriers of the APOE4 allele would show difference specifically in default mode connectivity. And so before I move to the results of this study, I just want to focus in on this key difference between what we were just talking about, which is functional connectivity in the context of a task, and what we're talking about now, which is functional connectivity in the context of a resting state. So the key difference is that now we're interested in these spontaneous fluctuations in the bold signal, as opposed to changes caused by a particular task. So in the paradigm we were just talking about, you might ask, is A in this figure more correlated with B during a particular phase of a task or during a particular, when you're doing a particular thing? But for resting functional connectivity, you're interested just in to what extent do A, B, and C, or any pair therein, have similar spontaneous bold signal fluctuations. And it was asking this question back in the early 2000s that led to the discovery of these intrinsic connectivity networks. And so I already mentioned the default mode network shown here in red, but there's also well over a dozen now kind of well-established intrinsic connectivity networks, only 4 of which are shown here. This is a paper that was published actually just end of last year in Science that if you haven't read, I would encourage you to read because they took a very novel approach using the Allen Brain Atlas data and actually looked at genetic expression across these different intrinsic connectivity networks and found that genetic expression was actually pretty similar. So these regions are also not just functionally connected, but they're actually coherent in their genetic expression. But they focused in on that paper. And these 4 networks, because they are bilateral, so a lot of the networks break up into left and right, and they are robust and reliably can be identified across participants. So I chose to look at 3 of these. I was not interested in visual spatial, and we'll move on and talk about the DMN salience network, which is important for identifying emotionally and behaviorally salient stimuli in your environment, and sensory motor network, which is the combination of the main sensory and motor regions. So a little background on why we were interested in this age group and in the default mode networks especially. There was this paper that was published in 2009 where they looked at young APOE4 carriers and non-carriers. They defined their default mode network as you see in A, and then they found these differences where carriers were hyperconnected in various regions of the default mode network. And this paper has been cited well over 500 times, and is sort of has become the de facto ground truth when you ask about the effect of APOE on default mode network early on. And this is really important that we get this right because default mode network is one of the earliest networks that breaks down in Alzheimer's disease. It's actually affected in a lot of different disorders, and that's a whole other can of worms. But it was basically, in this study, there were only 36 subjects, and they ranged in age from 20 to 35. So we aimed basically to set out and ask this question in a more robust cohort. So we collaborated with Ahmad Hariri at Duke. He has a really amazing data set. Actually, he was very smart and years ago partnered with 23andMe to get whole genome data on his people because he didn't want to limit himself to a set of genes he was interested in. So they actually have in these subjects Alzheimer's genetic variants genotype, which is actually very rare in this age group. So he was able to share with us 601 8-minute resting state scans. And from that, we were able to get 144 carriers and 427 non-carriers. And these groups, it's actually pretty amazing, are really well matched across everything that we have on them. And I just want to point that out now because anything that we find that's different in these two groups is pretty remarkable. So the allelic distribution is exactly what you would expect based on published frequencies. And we took a couple of approaches here in this data set. The first one is called seed-based correlation, where you take a region of interest, in this case the posterior cingulate, and you look for regions across the brain that are functionally connected, significantly functionally connected with that region. And so what we're able to do here is essentially pull out this intrinsic connectivity network. And here you can see in B, C, and D, that is the default mode network we were able to find in this data set. And it looks looks exactly how we would expect, and we were pretty happy to see that. You can then do a simple t-test where you compare the different default mode networks that you pulled out with that seed in your different subjects and look for differences in connectivity. So when we did that, looking at non-carriers greater than carriers, we were amazed actually to find that that PCC seed, there are two clusters, both located in medial temporal lobe, where connectivity between PCC and medial temporal lobe was lower in carriers than non-carriers. We also looked at the opposite contrast and found no significant differences. This is just showing the same clusters from an axial view where you can see how well they track with the longitudinal axis of the hippocampus. We then repeated this procedure using the seed-based method in salience network and sensory motor network. This is a summary of the results, but I don't want you to try to take anything away from here. We'll go through them in a second. The main thing I want you to show you here is that all of our results were in the direction such that carriers had lower connectivity than non-carriers, except for one edge in the salience network where carriers actually have stronger connectivity. So if we step through that, I mean, this is what I just showed you in that first cluster, where you have decreased connectivity between posterior cingulate and medial temporal lobe in the carriers. And then we also found decreased connectivity from sensory motor to pre-SMA, sensory motor to anterior insula, and frontal insula to sensory motor, which is picking up the same signal most likely. And then we found increased connectivity in the carriers in the frontal insula to PCC, which is really interesting because that's actually linking probably the two strongest nodes of the salience network and the default mode network. And I have my ideas about why that might be. But while we were looking at this, we were trying to make sense of this group of findings. And when we started looking at them from the vantage point of task negative and task positive, if you label them as either task negative TN or task positive, you can see that the pattern is mostly this reduced connectivity between task positive and task negative regions in the carriers. So to provide support to that, or to serve as a set of parallel analyses, we took another approach and used a previously published functional node set. So this, Power and colleagues in 2011 published a set of spatially and functionally independent regions of interest in the brain and then assigned them to different intrinsic connectivity networks. So in this image, red is default mode network. And then we actually were just interested in nodes that were assigned to cognitive networks, so we took out any primary sensory network regions and continued on with 135 nodes. So before we asked the question, talked about default mode network in this set of functional nodes, we wanted to make sure that we were talking a little bit at least about the same thing as we were in the first set. So I just plotted the nodes from the default mode network that I just showed you in red over our default mode network that we got from the seed-based analyses, and we found that there was a 98% overlap, spatial overlap of our nodes. So that was at least encouraging that we're sampling the same regions of the brain. And the first thing we did with this node set was we looked at intra and inter-network connectivity. So when I mean intra-network connectivity, if this is our very simplified network, each of the edges has a weight based on their correlation coefficient, and the average is just straight up taking the average across it. If you want to look at intra-network mean connectivity, you're going to have to take each node from network A and calculate the correlation to each node in network B, and then repeat that and take the average. So in our data, we looked at first intra-network connectivity to see if there were any differences in how the DMN was interconnected, like they found in the other paper, and we didn't see any differences. But when we looked at intra-network connectivity, the task-positive networks versus DMN, we did see a significant difference, such that the carriers had greater, more negative connectivity. So there's a natural opposition of task-negative and task-positive networks in normal, healthy brains, and we're seeing that this is actually exaggerated in our carriers. We also looked at some graph theory metrics. First, clustering coefficient, which is just a way to measure how interconnected a node's partners are. So you're essentially counting up how many complete triangles you have. So if we drew a line between the leftmost nodes in this graph, our clustering coefficient will be 1, and as you take them away, you get a number less than 1. And carriers had higher clustering coefficient than non-carriers. This is a subtle effect, especially in the magnitude of the number, but we actually had half of this data early on and then repeated all of our analyses once we added 300 more scans. And this effect was present in both of the subsamples, so we feel pretty confident about it. We also looked at modularity, which is essentially just a way to look at how willing subgroups of nodes in a network are to congregate together. And again, modularity was slightly higher in carriers than non-carriers. So both what I showed you on the last slide and this is supporting this idea that task-positive and task-negative networks are further segregated in our carriers compared to non-carriers. If we look at just like the modularity spring plot, so if we just plot the 135 nodes that we're looking at based on their module assignments after running the Louvain modularity algorithm, they look pretty much identical, and they fall into modules the way we would expect a healthy brain to, with the DMN being segregated from the task-positive networks. So even though we see a slight difference in modularity when we look at the raw Q value, we can't see a difference with the naked eye if we just plot out the nodes based on modularity. And this, again, I think is just encouraging that these really are healthy brains. They really do behave the way we expect. That there are these differences that are robust, and honestly, I've done graph theory analysis on many data sets now, and you really almost never find anything. So it's just very interesting that this kind of all has come together to tell this one story about task-negative and task-positive networks. So our large cohort of almost 600 people is the largest ever to be used for a study of this kind, and I think that provides a little more weight to our findings. And then, of course, the behavioral consequences of these differences, which would be everyone's first question, is it better to have more segregated task-positive and task-negative networks? Is it better to have your default mode network pushing down those task-positive networks better when you're at rest? Are currently unknown, because these groups are so well matched on every test that we can give them that there's no variance, there's nothing to test. And if you look at, I have looked, there's no association between these different measures and their behavior. And this leads me into something that I think about a lot, which is the challenges with interpretation in neuroimaging genetic studies. So when you define risk for disease by the genetic risk score for a particular allele, and you find differences between risk and non-risk group, how do you interpret those findings? You can say that maybe it's early changes related to the disease, that you're seeing pathophysiological changes that are going to one day lead to full-blown clinical disease. You could say that your differences are an example of antagonistic pleiotropy, where a genetic risk factor that confers risk late in life must have some sort of benefit early in life to be selected for in that way. You could say that your differences were compensatory mechanisms, that here's this group of people that has this really bad risk variant, and so they have this compensatory activity in the brain that's allowing them to behave similarly to the people who don't have that risk factor. Or you could just say these differences are neurodevelopmental, that having a particular risk factor for a disease may have neutral neurodevelopmental effects that have nothing to do with the risk for the disease. So it becomes a little complicated, but in the study I just described to you, we can basically rule out preclinical pathophysiology, right? Because these people are 18, 19, or 20 years old. But if I had described the same results in a group of people who were 75, it wouldn't be so easy to rule that out. So basically, the point I'm trying to make is that in order to really do the work that needs to be done, which is focusing on preclinical Alzheimer's disease and understanding risk for the disease in that preclinical phase, we need to really better understand risk factors for Alzheimer's disease and also for any disease of aging across the lifespan and how that actually manifests. And another kind of way to drive this home is to show you this image from a paper that was published in 2014 out of Eric Ryman's group. This group scanned 60 infants aged 6 months to 22 months and found these robust differences in gray matter density based on ApoE genotype. So again, what does this mean? Maybe nothing, but it's there, and we need to understand it if we're going to ever understand the differences that we see in older adults where we really care to know what's going on. So finally, I just have a couple more slides. I'll talk about some implications of this work and future directions. This is like my favorite new image of 2015 from Alison Goat, who's amazing and basically the foundation of the Alzheimer's genetics world, and does a great job of capturing how rare or common these different risk factors for Alzheimer's disease is, the effect size of the risk factors, and then also the heterogeneity of the different molecular pathways that they're involved with. And I think this is just, I wanted to show this as just to say that we need to continue to work really hard to capture the clinical and translational utility of all of this knowledge as we learn more and more about the underpinnings of Alzheimer's disease genetics. And then this is a flowchart that I thought was a good sort of alternative to the one we showed you in the beginning, where I talked about how neuroimaging genetics methods are important to the prevention of Alzheimer's disease. This has, on the far left, the discovery of loci of genes that are associated with the trait, and moves all the way over to identifying patients who are most likely to benefit from a given therapeutic strategy. And neuroimaging genetics has an important role to play in both of those steps. But of course, there are the steps that link them, including identifying causal genes, which is the current bottleneck in this flow chart. And then after that, understanding the mechanism by which that gene acts, or the product of that gene acts, and then based on those mechanisms, developing therapeutic hypotheses. And so really the full picture understanding of all the different people who are contributing pieces of this puzzle is crucial to disease prevention. And one way that the kind of emerging acceptance of that idea is playing out today is that the very first clinical trial for a amyloid antibody, which have all been tested in trial and failed across the board, there's currently a trial where they are only enrolling people in the treatment arm of the trial if they are amyloid positive based on a PET scan and over the age of 65 and cognitively normal. So they're finally marrying what has been learned from these imaging genetic studies and what the bench scientists are doing in developing their antibodies. So I'm going to end today on a little bit of a newsy note. This is just a plot that I liked where it shows the percent change in frequency of various age-related causes of death between between 2000 and 2013. And you can see that from breast cancer to heart disease to stroke, we've had decreases in the frequency of these various maladies, and a huge increase in the number of people who died due to Alzheimer's disease. That number is inflated a little bit by doctors being more encouraged to list Alzheimer's disease as the cause of death when it actually is. and also by the fact that as people avoid these other age-related causes of death, they're more likely to be affected by Alzheimer's disease and die from it because there's no treatment. It's a very expensive disease. We'll spend over $1 trillion on it by the time 2050 rolls around. And this year, Congress passed a budget that was favorable for the NIH in general, but also had a big boost for Alzheimer's-specific research funds. So at least we are moving in the right direction. And with that, I'd like to thank the research participants, Susan and the whole Bookheimer lab, Ahmad Hariri at Duke, who's been really great and an amazing collaborator, my committee, and my other mentors who allowed me to do great things that are not research during my time at UCLA and that I've really enjoyed. And then my funding sources, of course, and last but not least, my cohort. Each of these people have done really, really amazing work during their time at UCLA and have provided to me like an invaluable support system and network during my time here. This was taken at this is the first picture of us all taken together at like our neuroscience retreat or something, I think, right after we got here. Thanks to Esther and Rachel for finding this. Okay, then I'm done. Thank you, guys.
Thank you for a wonderful talk. I'm sure there are questions. Yes. Do you find a difference between coronavirus and
Sometimes, but the only genetic risk factor we study in isolation is ApoE, and E4, E4s are so rare that it's really hard to get a big enough group of them. So we almost never look at them in isolation like that, because in a group of, so in our group of 571 young adults, I think we had 20. So we can't, it's hard to look at, we really just don't have the power to look at it yet.
have you found the people with the risk factors, especially the young ones?
Well, so the young ones we didn't have the recruitment strategy for that. I'm pretty sure they're like psych students at Duke who get extra credit for coming and doing this study. So the frequency in that study was just that is just population frequency of those different variants. For the studies that we've done at UCLA, we used a particular recruitment strategy where we recruited from these so-called worried well groups. These are groups of people who feel that their memory is starting to go, but they are clinically testing normally. And then also from support groups for people who are caregiving for their parent, who have caregiving for their parent. And so we know they have a family history for the disease, and if you enrich your sample for family history, you also enrich for all of these particular genetic risk factors.
I'm really fascinated by the idea that you're coming up with these testable hypotheses about nodal interactions that would be presumably facilitated by anatomical nodes. And is there any hope in the near future that the resolution of the anatomy can match, functional MRI data that you collect?
Certainly in primates, there's been a lot of track tracing that you can make your best sort of interpretation of what you're seeing with the functional data. But in terms of human mapping those connections, DSI and these different diffusion techniques have come a long way, and they're actually pretty high resolution now, and are able to map crossing connections and have overcome some of the limitations that were there initially when DTI was first being published and used. But I think so. There was a study that came out in Brain like 4 months ago that tried to take these the kind of breakdown of these networks a step further in interpretation, and presented it in this idea of the homeostatic balance between all of these networks, especially in this task positive and task negative, in the balance across those networks. And as you look and see differences in one edge, you could stop there, or you could try to understand how that fits into the whole picture. And they showed that in increasing severity of disease, as you get more and more disruption, more abnormal network balance, it's a cascade, that you actually see something happening here, and you have an equal and opposite effect in another connection that sort of is trying to balance out this breakdown. And so I like to think about it like that, and in the results that I showed you where you have that one strong connection across default mode and salient network amongst all of these weakened connections. It was very interesting that I did that work shortly after I read that paper, and it sort of is this idea that, again, these are all healthy people, right? So maybe if they have this phenotype where they have this greater segregation across networks, there has to be an edge that is going to kind of make up for that and create, and bring those networks back closer together. But that's totally theoretical.
I'm curious, when you're collecting this type of data, are people previously aware of this, or once you have it, how do you handle this? How do people handle it? Do you share it with them?
So now in that study, that clinical trial I described, they're actually part of the clinical trial is they're doing a study on whether or not it is beneficial to give people that information, especially about their amyloid positivity. So these people need to know that their PET scans are amyloid positive because that is the criteria by which they get into the study. So knowing that you have kind of critical mass of amyloid in the brain, even though you're healthy, like what does that do? How does that affect the lives of those people? So that's part of what they're actually doing in that study. Is this a feasible type of way? Is this a feasible way to study this, or is it actually going to cause undue harm? In terms of what we do, we never reveal their genetic risk status, and that's part of the study. When I was more participant-facing, when I was at Northwestern, I would get asked that question a lot, and we would just say that there's no medically actionable plan after receiving that information, so it's not, the NIH doesn't allow us to give it. And that means people were okay with it.